All right, so welcome back to the post-lunch session. So we're about to start, and Pranay Goel would be the session chair. Before handing it over to Pranay, I just wanted to say a few words because, in the hurry for, getting the thing running in the morning, I wanted to say a few words about the Institute. So the Institute of Mathematical Science was set up in 1962. So in fact, we're celebrating our 50 years this year. So the Institute is formally an autonomous institute under the Department of Atomic Energy. It's a fairly small institute, but we are one of the largest ones in terms of the faculty which is devoted to pure theoretical research. So the entire institute is theory-oriented. We don't have any experimental labs as such. The faculty is divided into three informal groups. So there's the theoretical physics, which is the largest grouping of all. It has about 36 faculty members. The rest of the faculty is kind of divided equally into maths and theoretical computer science. So the research conducted here spans a wide range from very pure mathematics, number theory, algebraic topology, and to cryptography. In physics, we have traditionally strength in high energy physics and string theory, but lately we have been moving a lot more into biological physics, social economic modeling, and multidisciplinary approaches. Theoretical computer science has also been extremely strong, with theory of algorithms, computational complexity, and logic being some of the stronger points among our faculty members' research over here. So that is a very short intro to our institute. And I thought I'd just ask Srinivas, my co-organizer, to say a few words about the IIT, which is like co-organizing this venture, and then give this mic over to Pranay to conduct the session.
Thank you, Sitabra. IIT Madras is, as probably a lot of you know, is one of the older IITs in the country, and there were 5 older IITs, and IITM is one of them. There are 15 of them, and you lose track of where they are and how many they are, and so on. And we have a huge expansion of faculty strength. Now we have about 500 faculty in the institute, and it's one of the prettiest campuses probably in the world. It looks more like a forest than a college campus. And the day after tomorrow's sessions will be conducted in IITM. So I'm sure you'll enjoy the sights of deer, and just stay away from monkeys. And of course, certainly stay away from the snakes. We have lots of them. So I hope you'll enjoy.
Good afternoon. My name is Pranay Goel, and I'll be your chair for the afternoon session. We'll begin with a talk by Vidya-mitra at the Center of Neuroscience, Institute of Science, Bangalore. And he's going to talk to us about brain mechanisms controlling decision making and boredom.
Thanks for that. Yeah, first up, I'd just like to thank Sitabra, Srini, INCF, OP for putting this together. There are not too many neuroscientists in India, and so it's always nice to be able to be in a gathering like this and talk mostly neuroscience. Yeah, so before I sort of dive into the talk, I just thought I'd sort of give a sort of general overview, a conceptual overview of the sort of approach I take to study the brain. And so the general question is to understand how our brain controls our actions. And I sort of approach them from actually at 3 levels. At the level of behavior, what we do is we ask subjects, typically human subjects, to perform novel tasks. And by observing their behavior, we try to actually understand the computations that enable this behavior. At the systems level, we try to understand the nodes, the various sort of nuclei that actually give rise to that behavior. And this typically actually involves either microstimulating the brain in primates, using transcranial magnetic stimulation, or actually looking at instances where some of these nodes are actually disturbed, as in Parkinson's disease. And then finally, at the level of single neurons, we can actually train monkeys to perform similar kinds of tasks and then record from single neurons to sort of relate the pattern of activity of these neurons and how they're integrated in the system, and that performs a computation. Now, the behavior that I actually study is the ocular motor system, arguably the most important part of the motor system. And what we do is, why do we study eye movements? Eye movements are a good sort of and a simple system for a number of reasons, and I'll just actually cite two. One is that a class of eye movements called saccadic eye movements can be willed. Okay, so you can instruct subjects and then they can perform these movements, and so that's sort of a necessary ingredient to study voluntary control. Secondly, and that's shown from this very old slide from Yarbus, eye movements are cognitively penetrable, and I think that idea was brought nicely by Bapiraju's earlier work, showing, for example, how the ocular motor system can do implicit learning. So this is a slide from Yarbus in which he was among the first to actually record eye movements of subjects performing tasks. And here you can see these fixations, okay, the brief pauses in the eye movements. So actually vision is typically an active process, involves eye movements. And so what you're doing is you're bringing the fovea, or the center of vision, to points of interest in the visual scene. And so you can see that the distribution of these fixations are not random, right? They're not just randomly sampling the image, but rather they're focused on points of saliency, okay? And that's an important aspect of eye movements. They're goal directed. These goals are essentially two types. Goals that are bottom-up driven. They're driven by the salience of the image. So if you have a bright spot or of light, your eyes will be directed, in some sense controlled by the image itself. The eye movements are also controlled by the subject. Okay, that is, they're driven in a top-down fashion by instructions. And so in this particular slide, you can see that the fixations tend to be on the sort of the faces of these subjects. And so typically in this, I think this particular experiment, Yarbus instructed his subjects to determine the age of subjects. If the instructions were to determine, let's say, the wealth status of these subjects, you'd find that the fixations would be around, for example, their clothes or items of jewelry. So you can see that eye movements, although they're very simple, they essentially sort of imbibe or express really a hallmark of decision making, which is that they're driven by both bottom-up and top-down processes. Now, this is sort of the model that I typically use, the rhesus monkey brain, and sort of the bare bones circuitry, the eye movement system, the saccadic eye movement system, is actually rather straightforward. It involves information coming from the retinal ganglion cells and impinging on a structure in the midbrain called the superior colliculus that has actually a motor map, as was described by Sten. And the output from the superior colliculus then goes to the brainstem, to the vertical and horizontal saccade generators, and then the motor neurons from the brainstem then move the appropriate muscles, so they yoke the eyes in a conjugate fashion, producing the saccadic eye movement. So typically, this circuit is actually very, very simple in a sense, and so it sort of involves an afferent pathway and an efferent pathway. And typically, you can actually measure the time it takes for visual signals to reach the superior colliculus by sticking an electrode and measuring the earliest latencies of visual responses, and that's typically about 60 milliseconds. You can stimulate the superior colliculus and evoke eye movements, and they typically get evoked in about 20 to 30 milliseconds. So essentially, you've got an eye movement that can, in principle, happen in 80 milliseconds. Okay, but it turns out that typically eye movements take a lot longer. They occur between 200 and 500 milliseconds. So not only are they much longer than they should be, but they're also very variable. Okay, so they can actually range from 200 to 500 milliseconds. And this sort of procrastination actually suggests an act of cogitation, okay, an act of decision making that involves a circuitry that is far more than just the afferent and efferent limbs. And so the idea here is that extra time is taken up by central decision making stages that involve, at least in the case of eye movements, at least two decisions: where and when you will move your eyes. For simplicity, I've shown them as serial processes, but they can be continuous or cascaded. And so in the next half an hour or so, I'll be actually talking about these aspects of planning that take place between the eye movements. Now, a particularly interesting area to actually look at sensory motor processing in the context of saccadic eye movements is the frontal eye field shown here, sort of embedded in the arcuate sulcus in the primate. The frontal eye field actually receives a lot of its input from both the dorsal and ventral streams of vision. And if you stick an electrode and stimulate with a small amount of current, you can actually evoke eye movements of a particular vector depending on where you're stimulating. Okay? So the input is vision, the output is a motor command. So it's really an ideal place to study sensory motor processing. If you now study and you record from single neurons in monkeys performing these saccadic eye movement tasks, and one such task is actually a visual search task that sort of tries to, in the laboratory, mimic the Yabus-like condition that is where you have typically numerous objects in the visual field, and then the decision for the ocular motor system is to actually select one of interest here. Okay, so this is actually a kind of again a combination of a bottom-up and top-down task. And so monkey fixates a spot of light, a visual search array appears containing an oddball target, and the monkey's task is to actually make an eye movement rapidly to the oddball, following which he gets a drop of juice. Okay, and the time between the appearance of the stimulus and the response is called the reaction time. Oops. So now when you record from a particular class of neurons in the frontal eye field, you get actually a characteristic activity, a class of neurons that are called visual neurons. They perform, well, visual selection. And I'll just take you through this slide. So when the monkey actually sees the search array, the target can be either in the cell's receptive field, or if the target is elsewhere, a distractor is in the cell's receptive field. Okay? So essentially you have two conditions when either the target is in the cell's receptive field or the distractor is in the cell's receptive field. And then you can actually record the activity from a single neuron in these two conditions and shown here. Okay, so this is the spike histogram of a neuron when the target is in the cell's receptive field. And if you superimpose these two conditions, and that's shown here, you get a very interesting phenomenon. So shown here is the time 0, time from the stimulus onset. And you can see that the initial visual response does not discriminate target from distractor. Okay? This is the time of visual encoding, as psychologists would like to put it. But over time, a selection process ensues that actually discriminates the target from the distractor. So this is visual selection. You can show that this visual selection, in a sense, conveys the decision of where the monkey is going to look well before he makes a response. You can show that it's actually independent of when he's going to look by training monkeys on a different variation of this task. In this task, you have monkey fixates. The search array appears as before. Now, following a delay called the target step delay, the target actually switches its location abruptly and randomly. Okay? And the monkey can do one of two things. It can compensate for the switch, okay, in which case it makes a saccade to the new target location, or it can fail to do so. It actually goes to the distractor location. So here you have a condition where for the same stimulus, okay, you have two different responses. You can actually disambiguate the visual from the motor end of things. And then you can record from these neurons and ask what happens to visual selection. And so this is, again, the same sort of visual selection neuron. And you can show that there is a visual selection process that occurs that discriminates the target from the distractor. When the monkey actually makes a response or compensates to this position, the new position of the target. What happens when the monkey actually goes away from the target? You can show that for these neurons, the selection actually occurs independent of actually the response of the monkeys. Okay? So since the stimulus is pretty much the same, the target is in the cell's receptive field, it will select. Nevertheless, there is a process that's independent of the decision of where to move that then determines whether the monkey will actually move to the target or not. These neurons, there's a separate class of neurons in the frontal eye fields called movement-related neurons that actually determine the outcome of behavior, and that's actually illustrated here. These neurons, unlike the visual neurons, are more tied to the behavior of the monkey itself. And so this is the data taken from Haynes and Scholl, in which they trained monkeys on an oculomotor task. And as mentioned before, monkeys show variability in the responses. Okay? So they have a reaction time distributions. What Haynes and Scholl did was they actually divided the distribution into actually 3 sort of bins. Those reaction times are the fast, medium, and slow. And then what they did was they asked what the activity of those neurons were for these 3 different classes of reaction times. And as you can see, the response associated for this particular neuron with fast reaction times show a ramping up that's actually rather quick relative to when the same neuron was associated with a medium and a slow reaction time. Okay, and for lack of time I haven't described this, but it turns out they also show that this ramping up occurs to a certain threshold level of activation, following which a response occurs. Okay, so it's as if these neurons are sort of accumulating the sensory evidence presented by the visual neurons shown before up to a particular threshold, following which a response is made. If these neurons actually convey the motor plan to the oculomotor system, then what happens when the plan actually changes or switches? And that's actually shown in this particular slide. So shown here is the accumulation in black, okay, up to the threshold for a neuron making a saccade into its response field. Okay, remember this is a movement cell. Now, on those trials in which the monkey compensated for the target switch, we could also describe the activity of the cell, and that's shown here in red. Here, because the response is away, he's cancelled this partially planned response and moved away from the cell's response field. You can see the cancellation actually in process. Okay, so this is what you'd expect of a neuron that actually is in some sense responsible for the outcome of behavior. These findings, along with a lot of sort of a fairly large body of work in oculomotor decision making, and I'm also including the work of Bill Newsom and Michael Shadlin in this, suggests a kind of a very simple model actually, which is in this particular instance is called a later model, and the idea here is that you can envisage a decision or a motor plan as a kind of accumulation of sensory evidence that's occurring over time. And when this accumulation reaches a criterion level, you get a response. And that's shown here. This model actually has stochasticity, so that not every trial is identical, and the stochasticity in a sense explains the reaction time distribution very nicely, which shows a rightward skew. Now this later model, although it's been a very popular framework to study sensory motor decision making, hasn't actually been causally tested. And so, and I'd just like to highlight that the following work is actually the work of my student Arjun, and well assisted by Ram. And this was his thesis work. And so when I say we, I actually mean he. So what Arjun first did was he asked whether accumulator models can actually causally account for saccade planning. Because remember, up to this point, by and large, it's recording from neurons, single neurons, and correlating their activity with behavior. But that's a bit different from causation. And so for this, what he did was he used microstimulation, which can actually evoke saccades when stimulated when in the frontal eye fields. And that's just shown here. So this is your typical saccade. This is a visually directed saccade to a target. I think I might missed a slide here. Okay, and that's how a typical saccade would look. Now, when this plan, the saccade is actually shown, when you microstimulate during the planning phase, you get something that looks a bit odd. So instead of actually going towards the target, okay, you sort of land up somewhere in between the target. Shown here in red here is really what is called the fixed vector saccade. Okay, it's the location, or it's the saccade that would have resulted just with pure microstimulation. Okay, so what you're seeing here is a sort of an average of the plan to this location and to this location. And so what we did was we made use of this idea of vector averaging to actually tap into the evolving motor plan as it was happening in real time. And the idea is illustrated here. So consider a motor plan in abstract terms as a motor vector, okay, as a population vector. And this has been sort of nicely shown by people such as Georgopoulos to be a fairly robust framework to explain motor planning in the motor system in general. Okay, so you're making a plan towards a target, and while you're doing so, you microstimulate the frontal eye fields, and then what happens is that you actually land up somewhere in between. It's sort of a vector addition or a vector average of these two responses. And then as the plan accumulates, in other words, as it increases, and that's shown here as the deviation from the fixed vector saccade, that's plotted here. And as this plan increases in strength over time, you'd expect that this deviation to increase. Okay, so the idea here is by measuring or tracking the deviation, you in effect can test the nature of planning as it's happening. And so when you do this for a particular session, you can see a kind of an increase in the deviation, a sort of a linear monotonic increase, at least up to a certain point, relative to the time, okay, of stimulation from target onset, which is sort of consistent with this idea of an accumulator. No, I think the slides have gone. And so as I said, the accumulator model has a sort of inbuilt stochasticity. So on some trials, you would have fast accumulation, and some trials you'll have slow accumulation. And then to sort of test this, what we did is we computed the slope, okay, for a given session, and then asked what reaction time was actually associated with that slope. And so consistent again with this idea of accumulation, you find an inverse relationship between the slope of the no-step deviation and the reaction time, establishing in some sense a causal relationship between accumulator and motor planning. So to just summarize, these findings suggest that the accumulator models can be used to successfully account for saccade planning. The next we asked, he asked whether saccade plans can be modified. And for this we actually we devised a task what we call a redirect task, which is a slightly modified double-step task. And so on a large fraction of trials, subjects, in this case human subjects, instructed to make a saccade to a target. This is a simple enough response. On a set of catch trials, 40%, after target step delay, a second target appears, keeping the first target on. And on these trials, the subjects were supposed to compensate. Okay? They're supposed to cancel or inhibit the partially prepared response and go to the new target. Sometimes they were not able to do so. This is an act of control, demands inhibition, and they make an erroneous response. They go to the initial target followed by a corrective response. And the nice thing about this behavior is that you can actually test two subjects and cheat, because it's a voluntary task and these are not trivial tasks. So if you can plot the probability of error as a function of target step delay, you get a function that is supposed to increase with time. And so this again makes intuitive sense, because as the target step delay increases, you're well within into planning the first response, since you're not able to cancel or inhibit the partially prepared response. And so you expect more errors over time. And this is sort of the data from 9 subjects that actually essentially verifies that the behavior is fine. Now we try to as I said, encapsulate this model in the context of an accumulator model. We essentially extended the accumulator model to account for voluntary control. And the idea is, again, very straightforward. An accumulator produces the saccade, the initial saccade. On the fraction of trials, on step trials, when a new target appears, we assume that a new process, abstract computational process called a stop process, kicks in that attempts to actually cancel this partially prepared response. And you can have actually two outcomes. If the finish time of the go process, in a sense, occurs earlier than the finish time of the stop process, it's a race, then you'll get an error shown here. If it occurs later, the stop process essentially cancels the go, allows a second accumulator to finish producing the corrective response. And so the stop process fundamentally partitions the no-step distribution into two daughter distributions, one that produces errors that are faster and one that produces correct responses that are slower. And you can actually test again. This model explains performance very well because as the stop process is delayed over time, you'd imagine that the finish time will also correspondingly be delayed. And so this partition will move towards the right, producing fewer and fewer correct responses. Okay? And so we've actually extended this model even further to account for stochasticity, not only in the go process, but in the stop process as well. And so the reason why we did this is we want to actually push this race model to the limit. Okay, we wanted to ask whether we can actually predict the time, the slowest erroneous responses, okay, that are possible. And these will be responses that actually occur here. Okay? Since this is actually 2 sigma, this should actually occur about 95% of the times in a 2-tail, or about, you'd expect a violation of about 2.5% on the upper tail. And so this is the expected distribution of errors. And we've taken a more liberal statistic given by this inequality, and it turns out that we could tolerate an error of about 12.5% on the upper tail. And it turns out that if you actually measure the errors, the reaction time of errors that violate this 12.5% criterion, you get a value of 10%, which is sort of well within this limit. So by this criterion, the race model seems to work. Now, we hope this slide works because it's sort of important. Now, what we had subjects do was actually the same task, but in the context of a saccade target that was about 30 degrees. Okay? What that actually does is an interesting thing. On a small fraction of trials, instead of producing a single saccade to the target, you actually you fractionate a single action into 2 movements, okay? Producing a primary saccade and a secondary saccade, okay? And that just happens spontaneously. And so now this gives us the opportunity to study control not only before before the action starts, but even after the action starts, at the point when the movement is happening, and at the point of the intersaccadic interval between 2 movements. Okay? And so then what we did was we used our race model to then test, okay, the ability of the race model to predict the percentage of erroneous responses. And shown here is a kind of response that shows a kind of what we call a midway correction. So the monkey, human subjects, graduate students, are doing these experiments, and you could see a stopping that's actually happening during the movement itself. Okay, so what this actually tells you is that these saccades, although they're thought to be ballistic, they, if given enough time, you can actually be reprogrammed in flight. And if you actually then ask, what the percentage of erroneous responses are, you get a value again close to 12.5%, well within the limits set by the race model. Interestingly, however, we find a class of responses that we call hypometric errors. Okay, and these guys look like this. Okay, they should have been stopped at some point here. Okay, there's an intersaccadic interval that's happening, but the subjects actually go through well with their movement. Okay, it's as if they had chunked the response, they reached a point of no return, and even though the stop signal should have actually, by our criterion set up earlier, should have stopped this process, was not able to do this. Okay, and here you find a huge number of errors, 78%, that are not actually being able to be explained by the race model. And this just sort of quantifies this idea here. So here the secondary saccards, shown in white here, are well beyond the stop to sigma criterion across all the subjects. And so to us, actually, this is evidence of a point of no return. And although this search for the point of no return has actually captivated psychologists and particularly people doing motor psychophysics for many years, decades, I think this is actually the first clear evidence of a point of no return. And so the idea is actually illustrated here, that while you're producing a saccade, you can stop at any time, okay, given enough time, and you can actually correct. But if you're either chunking or you reach a point of no return, where the amplitude at some point, you're really refractory to new inhibitory input. So even though the cortex is aware of the change in plan, it's somehow not able to convey this to the brainstem. Okay, and we're currently trying to actually look at whether we can actually find the neural correlates of this very interesting switching process that can cut the cortex off from the rest of the periphery. So can saccade plans be modified? Those saccade plans in general can be modified during the planning stage as well as during the execution stage. Secondary saccades of a multi-saccade gaze shift are not amenable to modification. Can we track the saccade plan as it changes? Again, we use the same task, the redirect task, to address this time in monkeys. And so what we did is we had the target step delay. This target step delay, as I said, it induces a change of plan in humans as well as in monkeys. And while the plan is changing, we could actually microstimulate at various time points, okay, following the target step. And again, the idea is really an extension of what I told you about. You initially expect a deviation towards the target while you're microstimulating. Then at some point, the deviation, there'll be an inflection, and it should cross over to the other point. Okay? This time of crossover is thought to reflect the change, the time when the actual decision changes in real time. And so we wanted to actually test whether the microstimulation can reveal that. And so this is the data from one session. So this is the deviation plotted. So shown here in green is the initial target, red the final target, and this here is this switch time, which corresponds to this zero crossing time. Shown here is a time of the evoked saccade. Now the evoked saccade actually happens before the switch time. What you'd expect to see is a deviation. As I said, this is the initial target shown in green, the final target shown in red. As I said, the microstimulation, maybe I've forgot to mention this, but that it actually happens orthogonal, okay, in a direction that's orthogonal to the initial and final target. So the deviation can be easily measured, as it's actually switching its plans. So if the saccade, evoked saccade happens before the switch time, you'd expect a deviation, okay, because he hasn't switched, towards the initial target. I've already told you about that. But what's actually interesting is that the corrective response actually also occurs to the initial target. You see what happens when the switch time actually occurs before the evoked saccade. Now, as you'd imagine, the plan, it's already switched. So now the deviation is now towards the final target. And correspondingly, the corrective saccade also occurs to the final target. Now, the interesting case is when the evoked saccade actually happens around the switch time. Okay, you're just catching the switch as it's happening. Now, you get an interesting pattern. The initial deviation, okay, is towards the initial target, okay, but the corrective response, okay, is actually towards the final target. So around this time, you just caught the switch, and then you can actually quantify this, and you can actually show that the crossover time this corresponds to the switch time. So we can actually track the time it takes to change of decision. We then asked whether we could use this to actually reveal the mechanism, the computational mechanism underlying a changing saccade plan. And the idea is, again, a sort of extension of what other people have been doing. And so what we do is we model accumulators corresponding to GO1 and GO2 process, planning a saccade to the first and second target as a drift diffusion process, the mean drift of mu and standard deviation of sigma, so that we add this noise at every time step. And that's as shown here. Okay. Now, changes of mind, okay, are not actually trivial for the brain. I mean, we do this all the time, but they produce a very interesting, well, in some ways a paradox, because you have an accumulator, okay, so this is the initial plan. It starts off earlier, okay, so by definition, the second accumulator, the correct plan, is much weaker. And in fact, if there is any inhibition of these two incongruent plans, this inhibition should be far greater of this plan onto this plan. Okay? In this scenario, you should never actually expect a change of mind. And so, as you'd expect, a go-go independent model fails to actually account for behavior. And so, this is actually the data from a given session. This is the deviation profile, okay, the normalized deviation profile. And as you can imagine, this model never actually predicts the compensation function. Okay, so what we then did is we tried to incorporate changes, okay, in these models by adding interactions, okay, between these accumulators to try to actually fit the data. In this process, what we did was fundamentally we only fit the behavioral, the compensation functions. We let the deviation profiles just as is, okay, and then we asked the ability. We just essentially asked, you know, which model performs better or the best. And so these are the modifications. We first added kind of inhibitory interactions, reciprocal inhibitory interactions between these two accumulators. In this case, you know, we had the coefficients were the same. So as you can imagine, it doesn't really change anything, and you get the same output. Then we made these interactions asymmetric. Okay. So what we did was we gave this guy a bit of a boost over this guy. So we made the beta GO2s, okay, the inhibition on from GO2 to GO1 much stronger than vice versa. Okay. And as you can see, this model actually did much better. But however, when we actually tried to predict the crossover time from the observed crossover time, there was a sort of an underestimation. So what we did was we further modified these models. We had an asymmetric and actually had different rates. This actually didn't help. You again had this underestimation. This was pretty good, but this was bad. We then actually added, as I talked to you about earlier, a stop process, okay, with its own drift rates, and we made the stop process inhibit the GO1 and GO2 symmetrically. Okay, so it's an agnostic stop, just inhibitory control. You cannot distinguish the first plan from the second plan. And this also did reasonably. It fit behavioral data. It also fit the deviation data, and this was a bit better. But there was also an underestimation. We then added asymmetric inhibition. So what we did was we had a spatially selective inhibition. We fixed the beta GO2 at 0. So the stop somehow was a bit smarter. It was spatially selective and only stopped the GO1. And what this turned out to be pretty much, you know, the best model, at least in our hands. And so to just summarize, you know, successful changes of plan may involve a spatially specific inhibitory process to suppress the preparatory activity corresponding to the earlier plan in order to allow the new plan to manifest. And so that pretty much summarizes what Arjun did over his many years. Thank you very much.
Time for a few questions.
Thank you very much.
The microsaccades that you saw in Jarvis diagram of Rippon's painting are what is the latency of the
You're talking about the secondary saccades?
yeah, the microsaccades that you saw in the line in Jarvis' painting that you pointed to. So you made small saccades.
Oh, you're talking about the breaking up of the saccades?
Yeah. That's entirely different thing. Or do you see macrosaccades?
Oh no, these are these are saccades. No, they're not microsaccades. These are saccades that are actually greater than, we had a criterion, I forget, but they're actually about, they could be 6 to 8 degrees. The original saccade, okay, the length was about, I think, 35 degrees. So it will be broken up into 20 degrees, 10 degrees, 20, So these were not microsaccades. They were actually regular saccades. Okay. So could go up to 10 degrees, yeah.
If not, let's thank Adi again.